Well, welcome everyone to the fifth in our series of our monthly MycoTalks. My name is Gordon Brown here at the MRC Centre for Medical Mycology at the University of Exeter. And my co-chair this month is Oliver Kurzai from the University of Würzburg. Now, before I start, I need to apologize for the email that went out about the timing start, but that was an error in the system. We apologize for that. Just to confirm that these MycoTalks will be held every month at 4 p.m. On Greenwich Mean Time. So that is firmly fixed. So we have two fantastic talks for you today. The first is from Professor Joe Heitman from Duke, who's going to tell us about RNA-dependent epimutations and antifungal drug resistance. And the second is from Tobias Hohl from Memorial Sloan Kettering Cancer Center, who's going to be telling us about the microbiome and pulmonary antifungal immunity. Now, for those of you who haven't joined these talks before, just to give you a bit of background, the purpose of these seminars really is to provide a platform in which the entire medical mycology community can share the very amazing variety of different research that is performed here. And so what we do is we have two talks every session, and we try and have diversity in these talks in terms of career stage, gender, and also specifically the field of research. Now, what will happen is that each one of our speakers will give a 30-minute presentation or thereabout, one after the other, and at the end we will have a question and answer session. And so if you want to pose a question to one of our speakers, please use the Q&A function that you will see at the bottom of your screen. This is very important because we're not monitoring the chat function. And then at the end of the talks, Oliver and I will do a sort of a tag team type of thing where we will submit the questions to the speakers. Now, there's been some question to us about how this question and answer session works. People are feeling that their questions are not asked immediately. It's quite a difficult thing to organize because we have a stream of questions coming in on the question and answer session. And what Oliver and I will do, we'll extract the questions to our speakers and we will try and combine similar questions together. So we apologize if your questions are not asked in as quickly as you may submit them, but we will try and get to them all if there's time. Also, I would very much ask that when you submit your questions in the Q&A session, please put the name of the speaker to which that question is being addressed. It'll just make it very easier for Oliver and I to then try and pull out the questions from this long list as I've mentioned. Now, also, we record all talks, so if you or members of your team are not able to see this session live, of course, you can find this recorded presentation on our website, and you can see the link at the bottom of your screen. Now, just again to remind you, or to let you know, that you need to register for each one of these MycoTalks. It's not a rolling registration, but you can get reminders and register again through our website, which is listed at the bottom there. And if you register for our MycoTalk session, you will get an email invitation that's automatically generated that will then allow you to register for each talk as they come up. And as you can see, we've got another range of very excellent talks planned for December and January as shown here. And then finally, before we hand over to our speakers, I just want to thank the team at the background here, Jules, Lorna, and Alberto, who take a huge load on running this talk. So just thanks to them very much. So I'm going to stop sharing now, and I'm going to hand over to my co-chair, Oliver Kurzai, who will introduce our first speaker today, Joe Heitman.
Yeah, thank you, Gordon, and welcome also from my side. It's a pleasure to be here again and share this session again with brilliant speakers. And it's a great honor for me to introduce the first speaker, and that is Professor Joseph Heitman from Duke University. And thankfully, he's a speaker that does not need a lot of introduction because I guess everyone knows him. He is famous for working on several fungal pathogens, Cryptococcus mainly, but also Mucorales. He also is linked to this topic that immediately attracts attention, which is sex in fungi. And that's always a reason to look forward to the talks. He's editor of many journals, has received numerous awards and distinguished lectures. But when I asked him what is most important to him, he reminded me of the Woods Hole Mycology course, where he is actually active for since 20 years. And I think that also shows that he's also a great teacher. And that's probably the best reason to look forward to his talk. Joe, thanks for making it. And I'm really looking forward to it.
Thanks very much, Oliver, and thanks to Gordon and Oliver and the other organizers for the invitation to be here today. And I'm going to open up my talk. Hopefully you should see now the first slide. And the story that I'm going to tell you today has to do with a novel mechanism of antifungal drug resistance involving epigenetic mechanisms. And I want to begin with a historic context. This is Max Delbruck, who many of you know was a physicist who transitioned to working in genetics and molecular biology in the early days of the phage school at Cold Spring Harbor and other institutions. And he's famous for this experiment conducted with Salvador Luria. It's in fact referred to as the Luria-Delbruck experiment or the Luria-Delbruck fluctuation test. And it has to do with the basic nature and origin of genetic mutations. It was published in the journal Genetics 77 years ago this month. It's a very simple experiment. They started with the bacteria E. coli, and they screened for resistance to a lytic phage called T1. And there were two arms to the experiment. In one, they took a single subculture, divided up into multiple aliquots, and challenged it with the phage. And you can see the appearance of resistant isolates with some variation. And this is just standard deviation around the mean for this experiment. But the brilliance of their experiments was to do a second arm in which they grew multiple independent subcultures. And these were challenged with the phage. And you can see that in some cases, there were no resistant isolates. In others, there were a few. And in some subcultures, there were many, many resistant isolates. And these are referred to as jackpots. And this is dependent upon when the mutation for phage resistance arises in the culture. If it arises early, you see a large number of resistant variants. And if it arises late, you see no or very few isolates. And by combining this experimental paradigm with some sophisticated statistics, they could infer that mutations were spontaneously arising in the culture before they were exposed to the phage selective pressure. Put another way, mutations arise spontaneously and then selective forces act upon them later for selection. Now, you may also remember that Max Delbruck and Salvador Luria shared the Nobel Prize in 1969 with Al Hershey for these discoveries. And Al Hershey is also immortalized for the Hershey-Chase experiment with Martha Chase, his colleague, where they showed that DNA in phages was the genetic material. Now, what you might not know of this history is that Max Delbruck switched to be a mycologist after he won the Nobel Prize. And he worked on a fungus that's part of this basal branch of the fungal tree of life that we now refer to as the Mucorales. He worked on Phycomyces blakesleanis as a model for how microbes can sense external cues. We spent some time working on Phycomyces when Alex Idner was in the lab, but then when Su-Chan Lee arrived as a postdoc, we pivoted to work on Mucor succinyloides because it's a dimorphic organism, it's a human pathogen, and because you can transform it and delete genes by homologous recombination. Now, both of those species lie within the Mucorales, which used to be part of the Zygomycete phylum of fungi. This is the fungal tree of life. We're all familiar with the Ascomycota and the Basidiomycota that are monophyletic. They have a common origin here. But these other phyla here are referred to as the basal fungi of the fungal kingdom, and there's much less known about these. That'll be the subject of our discussion today. Now, zygomycetes cause devastating human fungal infections. You can see some of those images shown here. These are devastating and can sometimes require surgery for surgical debridement. And our armamentarium of drugs is quite limited to amphotericin B and lipid formulations and two azoles, posaconazole and isobuconazole. And there's a diverse collection of understudied fungal pathogens that cause these infections. I want to introduce you to the organism that we're studying. This is Mucor. It grows as a hyphal mat on a standard Petri dish. And you can see it as a multi-branched hyphal organism in the microscope. And what we discovered early on is if we expose it to an antifungal immunosuppressive drug called FK506, it blocks this dimorphic transition and it grows as a yeast. You can see this multi-budded yeast in the microscope here. This was actually rediscovering this dimorphic property that had been discovered by Louis Pasteur in the 1800s, that under anaerobic conditions, you also enforce a multi-budded yeast growth. And the novel feature here is that this is under aerobic growth conditions that a calcineurin inhibitor blocks the dimorphic transition. We know a great deal about this drug, FK506. It's a natural product, a macrolide produced by bacteria in the soil. It enters the cell and binds to FKBP12 to form a high affinity complex that then binds to and inhibits the phosphatase calcineurin, has a catalytic and a regulatory subunit, and it's bound and activated by calmodulin in response to calcium. This is the ternary crystal structure, FKBP12 in gold, FK506 in blue, and calcineurin A in green, bound to the regulatory subunit calcineurin B. The inhibitor binds to this unique regulatory region of the phosphatase, not to the active site. And it's what makes this inhibitor exquisitely specific for just this phosphatase in the cell. And this mechanism of action is conserved all the way from the fungal kingdom to our own immune system. This is the mechanism by which this drug inhibits T cell activation and blocks transplant rejection. And it's the reason that FK506 and drugs in this category have gone on to revolutionize human organ transplantation. Another drug in this class is cyclosporin. It binds to a different binding protein, but converges to inhibit calcineurin, again by associating with this unique regulatory region. So to return to mucor, this is the wild type organism on YPD medium. This is the yeast phase grown on FK506. If we prolong this incubation, you see the appearance of these drug-resistant hyphae, and they're either arising early in the culture or late in the culture. We purified these resistant hyphae and then asked what's the genetic and molecular basis for their drug resistance. Now, as you might imagine, one category are Mendelian mutations in the known drug targets, FKBP12, calcineurin A, and calcineurin B. And some of these isolates are shown on these Petri dishes here. These are mutations in calcineurin B or mutations in calcineurin A that confer drug resistance. We can model these on the crystal structure to understand how amino acid substitutions on the target can block the inhibitor from binding. Those are dominant gain of function mutations, whereas the mutations in FKBP12 are recessive loss of function mutations. So this establishes that the drug is acting through the known drug targets, but it doesn't really teach us much more beyond what we already knew, that this was an inhibitor of this highly conserved phosphatase. But there's a second class of resistant isolates that I want to tell you about. And one such isolate is shown here. Here's the wild type and a resistant isolate, CS2-1. On FK506, the wild type makes a yeast, but this drug-resistant isolate grows as a hyphae. And it's cross-resistant to a second drug called rapamycin, which also binds to FKBP12, but instead of inhibiting calcineurin, it inhibits TOR. And the fact that these mutants are cross-resistant to both of these drugs implicates some defect in FKBP12 as the underlying mechanism of action. But when we sequence the gene, there are no mutations in FKBP12. There's no mutations in calcineurin A or B, and there turn out to be no mutations anywhere in the genome. So what is the basis for this drug resistance? When we performed a Western blot analysis with an antibody that cross-reacts with the mucor FKBP12, these drug-resistant isolates lack the protein, and they also lack the messenger RNA encoding FKBP12. So the protein and its message are both missing. At this point, the student working on this, Cecilia, came to my office and said she was very frustrated because these isolates of hers kept reverting to being drug sensitive on passage. This experiment codifies those conclusions. Here are two wild type strains. They remain as a yeast no matter how long you passage it. They remain sensitive to FK506. If we delete FKBP12 or have point mutations in it, these are stable Mendelian mutations that always grow as a hyphae, indicated as the H. These are three of these unstable epimutant isolates, and at some stochastic number of passages, they revert from being drug resistant to being drug sensitive and growing again as a yeast, That's shown on the slide here. They revert to being sensitive to FK506 and to rapamycin. And when that happens, the protein FKBP12 is now reappears, and the message encoding FKBP12 also reappears. So when they're selected to be drug resistant, the protein goes away. When they revert to being sensitive, the protein comes back. What could be the mechanism for that? Well, colleagues of ours in Spain, Rosa Ruiz Vasquez, Santiago Torres Martinez, Victor Gare, and their labs defined an RNAi pathway in mucor involving the canonical components, dicer argonaute and RNA-dependent RNA polymerase. So we entertained the simple model that perhaps RNAi was induced to silence the FKBP12 gene. When we made small RNAs from these isolates, from wild-type controls, and from their reverted sensitive isolates, we ran them on a gel and then probed with an antisense specific probe to the FKBP12 gene. You can see there are no small RNAs in the wild type, but in the drug-resistant epimutant form, there's abundant small RNAs to the FKBP12 locus, and they disappear when these isolates become drug sensitive again. We made small RNA libraries and sequenced the entire genomic repertoire of small RNAs, and you can see in a wild type strain, these are small RNAs corresponding to the 3 exons of the FKBP12 gene and the 2 introns. There's no small RNAs in wild type, but in the 3 epimutants, both the sense and the antisense exons are decorated by small RNAs, but not the introns. And when these isolates revert to being sensitive, all of the small RNAs disappear. Now, there are small RNAs against other loci in the genome, but there's no change in the abundance of those small RNAs across this isolate set. And you'll also notice there's no small RNAs to the neighboring gene on the 5' end, and not shown here, there's no small RNAs to the gene on the 3' end. So to a first approximation in these drug-resistant isolates, the only place you see an increase in small RNAs is to the FKBP12 gene. We went on then with Sylvia Kahlo, a postdoc in the lab, to interrogate which components of the RNAi pathway are involved. The experiment was to start with a wild-type strain or isogenic mutants lacking dicer, RNA-dependent RNA polymerase, argonaute, or other components, and then to measure the frequency of epimutants based on these small RNA blots. In the wild type, we see approximately 30% epimutants, and in the mutants that are required for the pathway, it's below the detection limit. So we can show all of these components listed here are required for the epimutation pathway. Similarly, we can exclude other RNAi components. For example, argonaute 2 and argonaute 3 are completely dispensable for the epimutation process. And then a third category emerged from this analysis that we had not anticipated. And these are mutants in RNAi pathway components in which there's an elevated frequency of epimutants and in which the epimutants are very much stabilized such that on passage, they don't revert when they're epimutant at the FKBP12 locus. There are 3 components in this category: RNA-dependent RNA polymerase 1, RNA-dependent RNA polymerase 3, and an alternative dicer called R3B2. So taking all of this information, we can put it into a model that posits that there are multiple competing pathways. In the cell, there's the epimutational pathway that we've been discussing that generates antifungal resistance. There's an alternative pathway that Victor Gare and his lab have defined that's a non-canonical RNA degradation pathway. And notably, the 3 components that compete for the formation of epimutations are unique to this pathway: RDRP1, RDRP3, and R3B2. When this pathway is mutated, it channels aberrant RNAs that normally would enter both pathways into the epimutation pathway, increasing their frequency and their stability. Now, we've uncovered the role for this pathway using mutants, but you can imagine there might be conditions such as stress or infection, which might modulate the levels of components of this pathway or of their activity to fine-tune entry of candidate targets into the epimutation pathway. And we'll return to that in a moment. So I want to summarize to this point what we found. Spontaneous FK506 rapamycin resistant mutants, they can result from stable mutations in the known drug target or unstable epimutations that lead to silencing of that same target protein. And in those epimutants, there's no nucleotide change in the FKBP12 gene encoding FKBP12. The mRNA and the protein are both absent, and there's abundant siRNA that are present. And this pathway involved may be regulated or it may be stochastic. And we say that it may be stochastic because we've tested a large variety of in vitro stress conditions and not found any that lead to induction of the pathway. But I'll show you in a moment some recent evidence that infection may be a stress that induces the plasticity of the pathway. And I won't go over the evidence, but the trigger for the formation of these epimutants appears to be a naturally occurring antisense message that we think forms a double-strand RNA and then is diced to produce the small RNAs to silence the FKBP12 gene. Now, we've gone on to generalize these findings in two general ways. The first is to show that epimutants occur in a second Mucor species. I've been telling you about work in Mucor lusitanicus. This used to be called Mucor circinoloides forma lusitanicus, but recently the phylogeny was clarified here. And a sibling species, Mucor circinoloides, in which we found epimutants at an even higher frequency than in the original species where it was first discovered. So epimutants occurring now in two distinct species of Mucor. Now, very recently, Grit Walter in Jena, Germany, along with Kirsten Voigt and Sibrin de Hoog and others, have defined the precise phylogeny for the Mucor circinoloides species complex. And you can see now that there are 16 phylogenetic species. We've been talking about circinoloides and lusitanicus. At least 7 of these are human pathogens. We're interested in extending this work now across the complex and have established a collaboration with Grit to bring these isolates into the lab and examine them further. Now, the other key discovery of this paper was the discovery of isolates that undergo a complete genetic cross in which the zygospores will germinate, shown in these images here. This is the first time anyone has ever seen the zygospores germinate, and it will allow us now to be able to do a complete sexual cross and ask whether or not these RNAi pathways are activated during sexual reproduction, similar to the finding that RNAi pathways are activated during sexual reproduction in Cryptococcus and also in Neurospora. Now, we've generalized our findings in another way, and that's to identify epimutations in 2 other genes. This is selecting for resistance to a drug called 5-fluoroerotic acid. And this selects for mutations in the uracil biosynthetic pathway, either pure F or pure G. In yeast, these are called ura3 or ura5. In this case, we could not isolate epimutants in a wild-type background. So the student working on this, Zanetta Chang, took a genetic helping hand and used the mutants that have a higher frequency of epimutants, RDRP1, RDRP3, or R3b2. And she succeeded in finding epimutants in pure F. These are the small RNA blots showing 2 such isolates, and also in pure G. Now, in marked contrast to the epimutants of FKBP12 in these mutant RNAi backgrounds, these epimutants are unstable on passage. So for example, here is her pure G epimutant, and you can see by passage 5, it's reverted to be sensitive again to 5-FOA. So we think that these 2 loci might be more recalcitrant to forming epimutations compared to FKBP12. They needed an RNAi mutant background to form, and they were less stable than epimutants at FKBP12 in this background. And it suggests that the relative propensity to form epimutants might not be uniform across the genome. It may need to be regulated for some loci in some conditions. Now, Zanetta also made small RNA libraries and sequenced those. These are the small RNAs across the pure F gene, which has a single exon, and for the pure G gene, which has 3 exons. And again, you'll see that these decorate the exons, but not the final experiments that Zanetta did that were reported in this paper asks two specific questions about epimutants during infection. Are they stable? If you select for an epimutant in vitro and then infect animals, will that resistance persist during infection? She recovered colony-forming units from the mice, and you'll see in all the peripheral tissues, liver, spleen, kidney, and lung, there was almost 100% stability of the epimutant. So we envision then if epimutants were to occur in response to drug treatment in either an experimental animal or in a patient, they could persist within the context of the infected host. Now you'll also note that in the brain, she did see about a 50% reversion of the epimutant. And so there's something unique about the brain microenvironment. Now the second experiment that she did was to take a drug-sensitive strain, infect mice, recover CFU from these same organs, and then ask, could infection induce epimutants in vitro? And for the liver, spleen, kidney, and lung, the level of resistance was similar to the infectious inocula. But from the brain, again, something unique happened, and about 5 to 10% of the isolates became drug resistant. And the vast majority of those are epimutants at the FKBP12 locus. So this is the first stress condition we found that seems to activate the pathway. It also seems to activate reversion of epimutants. So there's something unique about the brain environment. To follow up further on that, Zanetta infected mice, isolated colony-forming units day 4, day 6, and day 8 during ex vivo culture from the brain, the liver, and compared these to her infectious stock. And she made small RNA libraries from these isolates that have been through a course of infection in the animal. And remarkably, what was found in the data analysis by Carlos and Maribel was 7 loci in the genome for which small RNAs become much more abundant when the isolate has been passaged through the brain compared to the liver or the control. These define 7 genes, BEP-A through BEP-G. 6 of these are hypothetical proteins, but the BEP-A gene is either an ortholog or a homolog of a stress-activated MAP kinase, MPKA in Mucor, that corresponds to MPK1 in Cryptococcus. Now, plotting out those small RNAs, you can see they decorate the BEP-A gene, but not the neighboring phospholipase D gene. Importantly, Carlos and Maribel have been looking at RNA-seq analysis culturing Mucor lusitanicus in macrophages and found that some of these same loci have a decrease in abundance across the locus. And it suggests that these small RNAs might lead to a reduction in the message. Now, I'll note that these experiments were done in 2 different species by different people in 2 different labs. Maribel and Carlos just arrived in Durham at about 1 a.m. last night to start their postdocs in the lab. And so part of the plan is to repeat this in a controlled fashion to see if these small RNAs are leading to a concomitant reduction in the levels of the mRNA. Now, I'll also note that we have combined all of the CFU that we obtained at these data points. And so we don't know if this is a combination of 7 different epimutants or if these are isolates in which all 7 of these genes have been simultaneously suppressed. And we plan to repeat this experiment and then isolate individual CFU to repeat the analysis. We're also planning to delete these genes one by one to test the impact on the infectious cycle for the organism. Part of the plan here for Carlos and Maribel will be to look in detail at this brain microenvironment. And the plan is to take an epimutant or to look at induction of epimutants in macrophages and then in a number of brain reduction systems in 2 different blood-brain barrier models that have been developed by Yong-Sun Ban at Yonsei University in Seoul, Korea, to study Cryptococcus. So we've established a collaboration with Yong-Sun Ban to look at Mucor and his systems. We're going to collaborate with Debbie Silver at Duke to look in brain organoid models. She's a developmental and neurobiologist working on brain development. We want to take epimutants, look at their stability and induction in the brain organoid model. And then finally, we're interested in the whole animal model, but dissecting specific regions of the brain to ask if there's a unique microenvironment that we can distinguish in which these events are happening that could provide insights into the interaction with the host. So I want to summarize. We've shown that epimutations are general and occur in 2 related species of mucor. We want to extend this across that phylogenetic species complex. We found the epimutations now in 3 different genes. Epimutations can be, like mutations, spontaneous and stochastic. We didn't find induction in response to several dozen different stresses in vitro, but the experimental animal model infection seems to be a stress that's inducing epimutations and leading to their reversion. Now, epimutations can be strictly epigenetic. If we take those epimutants that have reverted and select again for epimutants in a second round of selection, they occur at the same frequency. So that provides evidence that there's not an underlying mutation in the DNA that's triggering this pathway. We also sequenced RDRP1, RDRP3, and R3b2 and didn't find any mutations in those genes in the FKBP12 epimutants or anywhere else in the genome. But I've also shown you that epimutants in the 2 peer genes occur in RNAi mutants that are disabled for the alternative degradation pathway. So you can envision both a genetic and an epigenetic step, and it suggests that the function of that pathway might be regulated under physiological conditions, such as infection of the brain in mice. So to close, from 1 genetic screen, we found 2 different kinds of isolates arising. One that's stable and Mendelian and has changes in the DNA that are permanent. The other is unstable and transient and epigenetic. And when the selective pressures relax, these isolates can go back to being completely wild type. And this allows the organism to sample transiently phenotypic space without making a permanent commitment to a change in the genome. And we think that this is interesting. And we think that there are logical consequences for human fungal pathogens, and that these types of pathways may be operating in other human fungal pathogens that have active RNAi pathways, including aspergillus, cryptococcus, Tayloromyces, and others. I want to close by thanking the members of my group that contributed to these studies. The epimutants were originally discovered by Cecilia Wall when she was a graduate student in the lab. And then Sylvia Kahlo, who had been a graduate student with Rosa and Santiago working on RNAi and mucor, came as a postdoc, brought all of her RNAi mutants, and then reduced this to practice. Su-Chan Lee was the first person to work on mucor in the lab, and he was instrumental in these studies. Zanetta Chang was the MD PhD student in the lab who contributed the analysis of the PRF and PRG epimutants and the in vivo analysis. And then Blake Billmeyer, who's now with Sarah Zanders at the Stowers Institute, was the bioinformatic muscle. This is a long-term collaboration with a series of investigators from Spain at the University of Murcia, Rosa and Santiago, and Curo. And then more recently, Carlos and Maribel, who were students with Victor Gare. And we've been collaborating at a distance with the 3 of them. And as I mentioned, Carlos and Maribel just flew in from Madrid via Dallas last night, and they're ready to start as postdocs in the lab December 1st. I want to thank all the members of my lab, acknowledge the NIH for their long-term support, including a training program that supports training in mycology that's currently in its 17th year, and acknowledge support from a Canadian program, the CFAR program, Fungal Kingdom Threats and Opportunities, for which I have the opportunity to serve as co-director with Leah Cowan at the University of Toronto. Thank you very much.
Thank you very much, Joe. And we have, I'm sure, 253 people who are all giving you a round of applause in their unseen rooms all the way around the world. Thank you very much for that. As I mentioned at the beginning, we are going to go straight to the next speaker, and we will hold questions at the end of the session. Can I just remind attendees, please put your questions in the Q&A session, and please title which speaker your questions are aimed for, because as I mentioned at the beginning, Oliver and I have to scroll through to find all the various questions. Thank you. Thank you again, Joe. And we'll move on now to Tobias Hohl. And it's my great pleasure to introduce Tobias. So he's one of those few individuals in the world who's actually a clinical academic in medical mycology. He hails from Switzerland and became interested in fungi following a postdoctoral period with Eric Paymer. He started his own lab in 2009 and moved back to the Sloan Kettering Center in 2013. And although he's only been running his own lab for about 10 years now, he's already contributed many seminal contributions to our understanding of the immunology of fungal infections. And today we're going to hear two stories from him, the first beginning with the role of the microbiota and ending with intercellular crosstalk in the lung as a defense mechanism against aspergillosis. So over to you, Tobias.
Thank you very much for the kind introduction, Gordon. And let me begin here and try and get my screen going. Okay. I hope everyone can see my screen now.
Yes, we can see it fine.
Great, thank you. So thank you very much for the organizers for giving me the opportunity to present the work of my trainees that's been going on in my lab now for quite some time. Just to introduce everyone, we work in New York City, and you can see here sort of the dense agglomeration of Cornell and Memorial Sloan Kettering, which is a dedicated cancer center, as well as Rockefeller University, where Joe did his PhD. And I just wanted to point this out because you see here the hospital complexes of Memorial. And in this hospital, we conduct about 500 bone marrow transplants every year, primarily for patients with hematologic malignancies. And these patients are highly susceptible to invasive mycoses and are really the inspiration for much of the work that we do. And to get everyone on the same page, I think it's important to understand the type of immune injury that occurs during a stem cell transplant. So humans typically undergo stem cell transplant for disorders of hematopoietic cells, particularly for acute leukemia or aplastic anemia, diseases in which just administering chemotherapy by itself is not a curative process. So in a stem cell transplant, patients get conditioning chemotherapy, which is often myeloablative. And that is followed about a week later by the infusion of hematopoietic stem cells from a donor. And then comes a waiting period until these stem cells engraft and are detectable within the circulation of that patient. And this pre-engraftment period is associated with significant injury, both to the mucosa due to chemotherapy and radiation injury to mucosal tissue, as well as to the host's immune system. And specifically, we see a significant elimination of the innate immune system. And these patients are susceptible to invasive mold infections such as aspergillus, but also to disseminated yeast infections from Candida, as well as bacterial and viral infections. Now, if a patient engrafts, there's a second period of potential vulnerability, and that is if a patient develops graft versus host disease, a process in which alloreactive T cells damage host tissues. We actually use calcineurin inhibition as a strategy to prevent graft versus host disease, and this really illustrates, the importance of Joe's work over the years. Starting with Mike Hall, but also understanding how fungi interact with this class of drugs. And graft versus host disease is treated with immunosuppression, often with corticosteroids, and this leads to a second period of vulnerability in these patients. And I'm going to introduce the two stories with two patients that we were involved treating at MSK. This is a 32-year-old man. Now he is actually doing quite well. He's followed up. He underwent a stem cell transplant in 2012 for acute myelogenous leukemia. And during his induction consolidation and his stem cell transplant, his course was complicated by a Candida albicans bloodstream infection, and he became quite sick. And what you're seeing here is a transverse section through this man's abdomen at the height of this illness. And typically the liver is a very uniform tissue. And what you can see here are these lesions throughout this gentleman's liver. And when we biopsied these lesions, we grew Candida albicans in pure culture. So he clearly had a disseminated candidiasis with end organ involvement. And so we looked at all the stem cell transplant patients among a 4-year period, around 800 patients at the time. And we found that we had an incidence rate of about 3% in patients in terms of invasive Candida bloodstream infections. This rate used to be much higher. It was around 15% in the 1980s. And then fluconazole prophylaxis came and really revolutionized the supportive care for these patients. So it would be unethical to perform a transplant in 2020 without giving antifungal prophylaxis. We use echinocandin drugs, specifically micafungin, because this allows us to rapidly get the calcineurin inhibitors at the right levels post-transplant. Unfortunately, azole drugs have drug-drug interactions with calcineurin inhibitors. And so that's why we use micafungin. And perhaps not surprisingly to the aficionados in the audience, we see quite a bit of Candida parapsilosis in our patients. Now, thanks to pioneering work by Mahmoud Ganoun, Ilyan Iliev, Julie Segre, and many others, we've begun to understand that fungi colonize all parts of the human body, not just mucosal surfaces and the skin, but we've come to understand that there is our typical fungal communities that we can identify, culture, and trace within the gastrointestinal tract, within the genitourinary tract, within really at all sites within the human body. What I just want to highlight here is that the community is highly site specific. So for example, the skin is colonized by very different fungi than the mucosal surfaces in the lung or within the gastrointestinal tract. And what we wondered was whether the microbiota, how that relates to these invasive fungal infections that we see in patients. There are really two models that we could think of at the time. One is that we could identify an endogenous source. And if that were the source for these invasive bloodstream infections, we should be able to find evidence for the same strains or the same fungal genomes at that endogenous source prior to their invasion in the bloodstream. A second alternate model is that there's an exogenous source, that these fungi are creating bloodstream infections because they arise from the environment. Keep in mind that these patients have indwelling vascular catheters, and there are many potential conduits for fungi to enter the vascular space. So we sought out to distinguish these models and to establish a robust microbiota analysis pipeline in stem cell transplant patients. And all the credit in the world here goes to Bingzai, who joined my lab after training with Xiaoran Ling, and Thierry Rolling, who provided really a lot of bioinformatic muscle to this project. And this has been a collaboration also with Nick Tassini, a technician in my lab, and Ying Tower, who's a bioinformatician and ID doctor at Sloan Kettering. And we wanted to know whether we could monitor these endogenous communities during the stem cell transplant process and to try and understand the relationship between intestinal microbiota and fungal bloodstream infections. A long-term goal of this project is to understand interactions between bacteria and fungi in the gut and to see whether we can identify states of dysbiosis that are highly relevant for clinical outcomes. And so I want to just tell you the first chapter in this story, which was published a little bit recently this year. And what Bing did is she went through our biobank. We've collected at this point around 18,000 fecal specimens from 2,800 patients over a 10-year period. And we can now go back and identify patients. This collection occurred prospectively And now we can go back retrospectively and identify patients with very specific clinical events. And really Ying Kaur established this unique resource at MSK. And we did a small nested case control study with 15 patients, including 8 patients who had Candida bloodstream infection. And you can see here in the gray rectangle, that is the time during which these 8 patients, the range of time during which they developed Candida in the blood. And what Bing did is when she cultured Candida from the stool of these patients, she could show that she could culture very high levels of CFU immediately preceding the period that these patients became candidemic. She could not culture CFU from these patients prior to the transplant day, and she could not culture Candida from patients who did not develop candidemia. Occasionally, we do see fungal colonies in these patients. Often these are Saccharomyces, which is likely foodborne, and none of these patients developed Saccharomyces bloodstream infections in the control group. And we can show this not only, we can show this, this expansion not only using culture-based methods, but also using bioinformatic methods. What I'm showing you here is 4 of these patients, and what you're looking at is the relative abundance of specific fungal taxa in the stool as the transplant process goes forward. And what you can see is that at the onset of the transplant, these patients have many colors in these individual bars. And this suggests a high level of fungal diversity at the onset of the transplant process, but those colors disappear rapidly. And you can see that prior to the fungal bloodstream invasion, you get domination by a single color up to a week in advance. And the species that is the dominant species in the stool always, in every case that we've examined so far, corresponded to the species that we then subsequently isolated by a culture-based method from the bloodstream. This looks quite different in patients that do not develop candidemia, and I've shown here 4 examples. These patients have much more diverse fungal communities in the gut throughout the transplant process. You're looking at a 40-day window here, which includes the period of engraftment in most cases. And you're also seeing, you do not see the domination by pathogenic Candida species. In these examples, you don't see it at all, but we rarely see it in patients who do not develop candidemia. And you can plot this here as a function of time. So if you look at the diversity of the internal transcribed spacer regions, it is much higher in patients who do not develop a candidemia compared to those that do. We've gone on to sequence several hundred patients at this point, and approximately 700 total samples. Cherry and Bing really wanted to answer the question, is Candida domination a requirement for bloodstream invasion? And in virtually every case it is. But we see bloodstream invasion in about 3% of our total patients. We do see Candida intestinal domination in about 15% of the samples, largely in this pre-engraftment period around between days 5 and about day 20 in these patients. And a current focus of our work is to understand whether this intestinal domination is relevant for other transplant outcomes, for example, the development of graft versus host disease, or the occurrence of non-relapse mortality in our patients. And so when we began this project, the way most researchers analyzed ITS1 data is to rely on operational taxonomic units as the basic definition. And so the way the OTU analysis works is that sequences that have less than 3% dissimilarity, so 97 out of 100 nucleotides are the same or greater, these were classified within an OTU. And in the bacterial literature, and for most fungi, this roughly corresponds to differences at the level of individual species. In 2016, Curtis Huttenhauer and colleagues expanded the resolution of amplicon-based sequencing and introduced the DATA2 pipeline, which gives us resolution down to an individual nucleotide. And that was very advantageous for us because we applied this to our results. And when and I'm just going to show you one example of how this has yielded biologic insight. So if we look at a patient with a Candida parapsilosis bloodstream infection, and we look at different Candida parapsilosis strains within the intestine, we can identify strains that have one of two sequence variants. We refer to these as assigned sequence variants 1 and 2. And in this 279 base pair amplicon, there is a single nucleotide difference that we can reliably identify in our immunogenomic analysis. And so I showed you earlier how an individual patient becomes completely dominated by Candida parapsilosis. But what I haven't shown you is that the strain that we find in the gut, whether it's the same strain as we can ultimately recover from the bloodstream. And so in this patient who underwent Candida parapsilosis domination 3 days after the transplant, we can see that that domination includes 2 assigned sequence variants of Candida parapsilosis. And over time, one of these variants becomes the dominant variant, ASV2. And on day 14, when this patient becomes fungemic, the isolate belongs to the ASV2 variant. Now, this was all bioinformatic until now in the intestine. And so what we did was we went back and we isolated individual colonies, fecal isolates from this patient. And when we sequenced these isolates, we could also see the shift in domination from the ASV1 to the ASV2 strains. Now, to get much higher resolution and to get more information, we were very, very fortunate to establish a successful collaboration with Geraldine Butler. And this work was really conducted by Geraldine and her outstanding graduate student who recently defended her thesis, Mihaela Ola. And what Geraldine did for us is she analyzed many, many different fecal and bloodstream isolates at the whole genome level. So Candida parapsilosis has a 26 megabase genome, and Geraldine also had access to a number of clinical isolates worldwide. And I'm showing this phylogenetic tree just to show you how much diversity there is within Candida parapsilosis isolates from an individual patient. I've shown you already that a patient can harbor ASV1 and ASV2 strains within the gut. And you can see that the intra-individual variability is greater than the variability that we see between isolates from different patients across the world. You can also see that in the cluster 2 isolates, we have blood isolates as well as stool isolates, but we were not able to resolve these initially. And so what we did was we took well-resolved SNPs across the genome and performed principal component analysis, which enabled us to look at the pattern of translocation of these strains from the gut into the bloodstream. And so I'll just show you the power of this analysis quickly in this patient where we have 14 isolates that Geraldine compared for us, 12 from the stool and 2 from the bloodstream. Importantly, all of these isolates belong to the ASV2 strains. And most clinicians would say that, you know, if you get a bloodstream invasion of a pathogen, and you can isolate that pathogen on consecutive days from the bloodstream, that this is a failure of an antimicrobial drug to sterilize the bloodstream. But what we found when Geraldine did her analysis, that thinking was incomplete. We found that the ASV2 variants in the stool segregated into 3 major clusters that she could resolve using the PCOA analysis, and that the 2 bloodstream isolates belonged to 2 separate clusters. And so what that's telling us is that the translocation from the gut reservoir into the bloodstream is stochastic and occurs repeatedly over time. Now, finally, just to give you some idea of what we're doing in terms of the bacterial fungal interactions, we found that stool samples with very high candida burden had a different bacterial environment than those with a low candida burden. Specifically, the bacterial density was much lower in those stool samples with a high candida burden, and the bacterial diversity as well was significantly lower. And so we wondered what bacteria are relevant for these observations. And we were very fortunate to be able to show our data and discuss it with Andy Coe at UT Southwestern. And he had found that a number of years ago in a really beautiful paper that specific commensal anaerobes, the Lactospiraceae and the Bacteroidetes, can mediate candida colonization resistance. And so the presence of these organisms can stimulate enterocytes to produce antimicrobial peptides that prevent candida intestinal colonization. And in our patients, we saw that in samples that had very high levels of candida, had very low levels of Bacteroidetes and Glaudia. And this was because patients were exposed to antibiotics with anaerobic activity, specifically the carbapenem drug class, which led to a reduction in these taxa that likely promoted the colonization resistance. So one area of focus for our group is to understand better these cross-kingdom interactions. So I just want to summarize and say that we can really trace candida and fungi within the intestine and identify states of fungal dysbiosis during stem cell transplantation. And this domination precedes stochastic translocation into the bloodstream. And here, Geraldine's techniques of whole genome comparisons have been incredibly valuable to identify the translocation events and steps that occur as part of candida pathogenesis. And finally, we are beginning to delve into cross-kingdom interactions between bacteria and fungi during this process. So I'm going to quickly pivot and transition to a second case, specifically to a patient that I saw in the ER a couple of years ago. And his main complaint was, "Doc, I can't breathe." This patient had graft versus host disease after transplant of the lung. This is referred to as bronchiolitis obliterans. And in the bottom right of the screen, you can see a chest X-ray in which the bronchiolitis obliterans is this very large airway that you see here. And this chest X-ray was done, or chest CT was done several months before admission. And you can see this new lesion here in the lung that has grown in the interim. And that lesion ended up being aspergillus fumigatus. This patient, unfortunately, ultimately succumbed to the disease. So we have been studying the host response to aspergillus for a number of years. And we've understood that myeloid cells are the major type of leukocyte that are important for host defense. So there are many indispensable myeloid cells for this process. In contrast, lymphoid cells, both in mice and humans, are largely can be compensated for. And mice that lack all lymphoid cells, including ILCs and NK cells, are not susceptible to aspergillus challenge. And so we've been working on this for a number of years, and really this has been a terrific collaboration also with Amaryllis Rivera. We know that neutropenic mice are highly susceptible, but we discovered a number of years ago, together with Amaryllis, that monocytes are also indispensable for host defense. And the way we did this was I generated a mouse in which one could deplete monocytes by administering diphtheria toxin without touching the neutrophils. And these mice, called the CCR2 depleter mice, when they were treated with diphtheria toxin, developed invasive aspergillosis and succumbed to the disease. And this defined monocytes and and monocyte-derived dendritic cells, which accumulate in the lungs of aspergillus-infected mice, as essential antifungal effector cells. And they do this in two ways. First of all, monocyte-derived dendritic cells can take up and directly kill aspergillus. This is a picture of aspergillus that is being engulfed, and the red dye is a viability fluorophore, and the blue dye simply to trace it. And so you can see the loss of the red dye indicates a fungal killing in these micrographs. But the other interesting observation that Amrelisa and I made was that in mice that lack monocytes, the neutrophils don't kill well. So if you look at the frequency of neutrophils that contain live aspergillus, which is indicated in the red gate, that frequency is significantly higher in mice that lack monocytes. And this is not due to a neutrophil recruitment defect. This is a functional defect in the neutrophil. And so we've been spending the last 4 or 5 years to understand the mechanistic basis of this neutrophil dysfunction. And so our original model when we started this work was that both neutrophils and monocytes infiltrate the aspergillus infected lung, and that monocytes provide an assist to the neutrophil. And this is work that was started by Shinji Kasahara in my lab and was recently completed by Yahui Guo, a postdoctoral fellow. And what Shinji and Yahui figured out was that a chemokine, two chemokines, CXCL9 and CXCL10, were dependent on the presence of monocytes. So in mice that lacked monocytes, because we had ablated them, the levels of CXCL9 and 10 were significantly reduced. That's shown in panels B and C here. And you can see also that aspergillus infection induces both of these chemokines in vivo. And we were very fortunate to collaborate with Andy Luster, who's also a Tri-I graduate. He developed mice in which the CXCL9 and CXCL10 promoters drive the expression of fluorescent proteins, so we can figure out which cells are making the CXCL9 and 10. And with this reagent, we're able to show that monocytes, monocyte-derived DCs, and to a lesser extent neutrophils, really drive the induction of CXCL9 and 10 during aspergillus infection in the lung. And this feature of monocytes and neutrophil is really triggered by the process of fungal uptake. So in this graph, in the blue and in the red regions, these regions indicate neutrophils and monocytes that are fungus infected compared to the black regions in which these are uninfected bystander cells. And you can see in these graphs that the CXCL9 and 10 expression is significantly increased when fungal cells infect neutrophils and monocytes. So this is a feed-forward amplification mechanism. Now, CXCL9 and CXCL10 signal via a receptor called CXCR3, and if our model is correct and these chemokines are important, then CXCR3 deficient mice should be susceptible to infection, which is exactly what Yahui and Shinji saw. So that gave us increasing confidence that this was an important signaling access during respiratory immune defense against aspergillus. Now, our model has been updated, and I didn't show you the data to show that a CXCL9 production is induced by signals through dectin-1 and card9 signaling, whereas CXCL10 is induced differently. It requires signals from type 1 and type 3 interferon receptor signaling. But the real puzzle for us, and this took us a year to figure out, was what is the target cell? Initially, we thought, well, neutrophils and monocytes, they probably interact directly with each other, but we could never find CXCR3 expression on neutrophils or on monocytes, whether they were isolated from the bone marrow, the lung, or the fungus-infected lung. So eventually we decided to search more broadly. And what we found was that plasmacytoid dendritic cells express quite a bit of CXCR3. And importantly, plasmacytoid dendritic cells are recruited to the lung in a CXCR3 dependent manner. The reason we had such difficulty figuring this out was that many preparations of collagenase that we use for digesting our lungs degrade an antigen on PDCs, and thus we were not able to see them until we used collagenase-free methods of making single-cell lung digest. So if this model is correct, then getting rid of PDCs should render mice susceptible to aspergillus. And Marco Colonna's group about 10 years ago generated mice in which you can very specifically delete PDCs by administering DT without touching the neutrophils or the monocytes. And these PDC depleter mice are highly susceptible to aspergillus challenge. And you can see that these mice develop evidence histopathologically of invasive aspergillosis. Now, what is happening mechanistically? So what we did was we infected these mice with our fluorescent aspergillus strain that allows us to quantify fungal uptake and killing by cells in the lung. And what we found was that the frequency of neutrophils that contains live aspergillus was substantially higher in PDC depleted versus PDC sufficient mice or control mice. In other words, the neutrophil, if you don't have PDCs around, the neutrophils don't work as well and don't kill aspergillus well. And one correlation of this killing defect is a reduction in the oxidative burst. So neutrophils from PDC depleted mice have a much lower oxidative burst than neutrophils from a PDC sufficient mouse. So we're currently working to understand how PDCs regulate the neutrophil oxidative burst. That is really at the forefront of some of the ongoing work in the laboratory. So just to summarize the second part here, we've identified a functional role for PDCs in host defense against inhaled Aspergillus canidia. Stu Levitz has done some very elegant work looking at human PDCs and their role in host defense against products of germination, against Aspergillus hyphae, and how hyphae might activate PDCs. But this work really emphasizes their early role against canidia. So with that, I am going to just want to thank everyone in my lab for their hard work and our funding sources. We currently do have positions that we would like to fill. So if anyone is interested, please get in touch with me. And thank you for this opportunity to present our work.
Thank you very much, Tobias. And again, you've got 250 people, I'm sure, giving you a round of applause. Otherwise, thank you both for your wonderful presentations. So we're going to open the questions now. As I said, please, attendees, please put your questions into the Q&A box and give the name of the speaker who you wish to direct your question against. And we're going to start with questions for Joe, and I'll hand over to Oliver who will start the process.
Right, thank you, Gordon. And Joe, I'll start by saying that there is a lot of compliments for your talk already in the Q&A, and I want to start with questions that target the question of how widespread the phenomenon you described is. First, there is a question with regard to if you look at different strains within the species, would all strains show these epimutations? And then how widespread would you say is it in the fungal kingdom as a whole?
Excellent question. So we've looked at different isolates within the same species, and there's definitely variability in the frequency with which epimutations are formed. But I don't think we've done an exhaustive survey yet to be able to say whether or not there's a difference, for example, between environmental and clinical isolates, which I think is an excellent question. So there's definitely variability within a species, and I think that's to be expected. We've found these so far in 2 related species of mucor. We spent some time looking in Rhizopus unsuccessfully and also in Cryptococcus, but I think there are many other things to think about in revisiting this. And some people have told me about unstable drug-resistant isolates they've seen in other species, including Paul Dyer and aspergillus. So my hope is that these findings will stimulate other people to look at other unstable mutants. Some of you may have seen a paper published in Nature from Robin Alshire's lab where he looked at unstable resistance to caffeine in fission yeast, Schizosaccharomyces pombe. And what he found there were epimutants that are dependent upon heterochromatin forming over the target genes, and it's also associated with RNAi. So it opens the door to epimutations involving RNAi or epimutations involving heterochromatin. And in plants and animals, we know of epimutations involving DNA methylation. So I think any of these are possible in the fungal kingdom. And my hope is that this will stimulate people to look more broadly through the kingdom.
Right, thank you. And the other question that is also coming up several times is about potential modulators of these epimutations. And this could be either external stimuli, like, for example, reduced oxygen, as you would have in brain tissue, or also intrinsic things like chromatin structure, and this could affect different loci and their frequency for epimutations. And can you comment on that?
So we looked at at least several dozen different in vitro stress conditions, and there's always more stress conditions we could explore. But to a first approximation, we didn't find any stimulation. And this was using toxins and inhibitors like rapamycin and cyclohexamide. And I think we also tested galvanomycin as an HSP90 inhibitor. The question of whether hypoxia might play a role is a very astute one, given that oxygen tension levels are different in different tissues. The one thing you need to disentangle, though, is that under hypoxic conditions, mucor is also stimulated to grow as a yeast, as it is under aerobic conditions in the presence of FK506. We never were able to see hyphal variants emerging under hypoxic conditions. And these epimutant variants that return to hyphae are not, for example, locked in the hyphal phase because they'll form a yeast under hypoxic conditions. But I think it's worth revisiting hypoxia as a potential inducing signal, maybe in the conjunction with those mutations that disable the competing degradation pathway. And my hope is that Maribel and Carlos will tackle some of these questions ahead.
Right, and one last question, if I may, that is the question, which genes in mucor are actually affected?
Yes, so I think potentially it could be all genes in the genome, similar to mutations. Now, there's always a bias towards mutations. Larger genes are more subject to mutation than smaller genes. Essential genes are underrepresented, for example, by serious deleterious mutations. So there's always some bias across the genome. The question of whether or not heterochromatin might play a role here, I think is an interesting one. We tend to think of heterochromatin as shutting down expression in regions of the genome. And it may actually be that heterochromatin could play a role by forming epimutations that are heterochromatin dependent. But the question of why the FKBP12 and the PRF and the PRG genes differ is a great question. Kaustav Sanyal's lab in Bangalore, India has just started doing H3K9 dimethyl chip across the genome. And I'm going to email them and ask them whether or not they think there's any difference across these 3 genes as soon as we finish. I think that's an outstanding question. And the question of which genes in the genome, there may be some that are more prone to this than others. There was also a question about whether or not the ability to form epimutants might be selected for in some way. And I think that's a very interesting question. You could imagine this being a regulatory process. And maybe those genes that were being subject to epimutation during infection of the brain, maybe that is under an adaptive evolutionary pathway for stress resistance and maybe in environmental predators that mimics in vivo infection conditions. It's a great question. I hadn't thought about it that way previously.
Okay, thank you. Gordon, do you want to continue?
Yes, thank you. So Tobias, there's been a couple of common questions and a couple of individual questions. I'm going to start with maybe 2 or 3 of the common questions. The first one is the possible environmental contamination of Candida parapsilosis. So I noticed also in your talk that when you looked at your species diversity, it wasn't present in the patients before they developed the disease. And Ted White has mentioned that it persists longer on surfaces. So it may be a environmental contaminant in these patients or a nosocomial contaminant. Have you considered that and looked at that at all?
Yeah, no, we thought about this possibility quite a bit. And you know, there's good data that Candida parapsilosis can persist in the environment. We have not found, we look for obviously for yeasts and molds in the environment, particularly when we see outbreaks in our hospital. We have not found a Candida parapsilosis to be a significant yield in environmental swab samples, but it does not preclude the notion that Candida parapsilosis may come from the environment. But what I think our data shows is that it initially establishes a reservoir within the gastrointestinal tract, wherever that original source may be. And that reservoir in the gastrointestinal tract has to be present in all the patients that we've identified in order for us to see a subsequent bloodstream infection. So we've never found a patient, and granted, you know, our numbers are fairly small, as where we have a Candida parapsilosis bloodstream infection in the absence of having a preceding domination event in the gastrointestinal tract. So that really argues that even if the fungus is coming from the environment, it is first establishing a niche within the gastrointestinal tract. We do see very small amounts of parapsilosis in the pre-transplant samples. You know, the way I displayed the data, it's hard to see things that are, you know, 1 or 2% abundance, relative abundance, but we do see that in the very early samples prior to receipt of the stem cells.
Okay, so related to that, then the other common question that came up with 2 or 3 people was they were wondering whether, knowing that this loss of biodiversity or increased, is there therapeutic options there? Fecal transplants, whatever.
So we actually, Ying Tower, my collaborator, has actually conducted a clinical trial that he's about to report the outcomes in. And what he did was he took patients with a healthy bacterial microbiota and banked samples before their transplant. And if patients lost bacterial diversity, he randomized them either to receive an autologous fecal transplant or not. And he published in 2018 that the transplant process works, that you can restore bacterial diversity using an autologous fecal transplant in the stem cell transplant population. This was a paper published in Science Translational Medicine in 2018. And the clinical outcomes paper is coming. I haven't seen the final data yet, but it does appear that giving a fecal transplant may have an impact on viral reactivation in particular in these patients. So somewhat unexpected. But we're currently exploring, you know, how, I think in the long run, the best way to do this would be to come in with a defined microbial consortium to restore either colonization resistance or to increase the diversity of the bacteria and fungi that are found in these patients during the transplant process.
Okay, I have more questions about the microbiome, but perhaps I'll hand back to Oliver for a couple of questions and we can pick those up in a sec.
Great. So what I would like to take up is the question with regard to the antisense RNAs, and where does it come from? How is it regulated? Why do you find it so targets in such a target specific way?
So for the FKBP12 gene, what we thought initially might be the trigger is that in the genome, there's the FKBP12 gene is convergent with the neighboring gene and the messages actually overlap. So we thought, aha, that must form double-strand RNA and be the trigger for the formation of small RNAs. But when we deleted that neighboring gene, it had no impact on the formation of epimutations at FKBP12. We subsequently found that there was a naturally occurring antisense to the FKBP12 gene that's just present in cells prior to selection. And both the sense and the antisense are destroyed during the epimutation pathway. In sequencing that antisense, we can see that it's derived only from the exons of the gene, not the introns. So it's not coming from transcription of the opposite strand of the DNA, for example. We think it's being produced by an RNA-dependent RNA polymerase acting on the normal mRNA to copy the other strand. We've tested the RDRP1, 2, and 3 individual mutants. They still all make this antisense, so there may be some redundancy for making it. We know that there's transcription from just about every region of the genomes for every organism that's been analyzed. There's lots of non-coding RNA, lots of antisense messages, and it's been sort of a mystery what all of this transcription might be doing. And in some organisms, it might be to provide the opportunity to form epigenetic events. So what we'd like to do is deep strand-specific RNA-seq for mucor under a lot of different growth conditions to ask exactly these questions. Where do you find antisense RNAs? How abundant are they? Do they change in response to stress conditions? And might that provide insights into which genes might be more predisposed to undergoing this epigenetic pathway?
Right. Another question is, given what you presented, one could assume that this strategy with epimutations and using RNA for short-term regulation is very efficient in adapting to stress situations. Do you think it's efficient in all situations, or could it also be detrimental to the fungus? Is it stress-specific or more a general phenomenon?
So initially, we thought that there might be a lot of collateral damage in the organism. So for example, if you silenced FKBP12, but you had turned on a kind of general pathway, maybe there would be dozens of loci in the genome that had been silenced. And when we analyzed the small RNA libraries, we were surprised to see just how exquisitely specific it seems to be. And I think that's because the isolates that grow out of these selective pressures have been selected. There may be other epimutations forming around the genome that appear and disappear, but if they're not adaptive, you won't see them selected for. Peter Sarkis from the UK published a really interesting paper on C. elegans recently, where they looked in experimentally evolved lines of C. elegans, but they didn't look for DNA mutations, they looked for epimutations. And they could see them appearing against many loci throughout the genome, appearing and then disappearing during the course of the experimental evolution. There was no selective pressure. And so their presumption is that these are occurring and then disappearing during the growth of cultured lines.
Okay, thank you. Gordon.
Tobias, so we have a couple of related questions about the microbiome. The first one is the oral microbiota, whether there's changes there that may influence what you see in the gut. And the second sort of related question is, do you see these fungal expansions in specific areas of the intestine?
You know, to try and get at that question, unfortunately, we were not able to sample the oral cavity together with the, you know, with the fecal samples. And so, you know, we do ingest quite a bit of, the diet is a really important contributor, I think, to the types of fungi that we see in the gut. I think it's important to keep in mind, though, that in the first 10 days post-transplantation, patients essentially don't eat because they feel absolutely terrible. They lose weight during this time. And we do more recently have started keeping dietary records of patients. And so hopefully in a subsequent cohort, we'll be able to look at this question more carefully. But I don't think that what we're seeing is just fungi that are passing through, because we can culture these at extremely high levels from the fecal samples. And food that patients typically can ingest, you know, it's like the autoclave food from the mice. It's severely restricted in terms of cheeses, for example, are not allowed. It is a low microbe diet. So I don't think we would be able to see these blooms, these culturable blooms in these patients, just based on any sort of dietary intake.
Okay, and then a question for me really that's related in terms of mice. If you look at mice and do something similar, do you see blooms in specific tissues, in specific parts of the intestine? Do you think
Yeah,
it's
so
possible?
Ilyan Ilyev has done some of that work where he specifically dissected out, the small intestine and different parts of the large intestine, and it's shown that the fungal communities do vary in a site-specific manner. That's not possible to do in stem cell transplant patients because we can't do colonoscopies on them. It's simply unsafe to do. These patients don't have platelets and would bleed in an uncontrollable manner if they underwent such procedures. So unfortunately, I would love to get more information on the mucosal, on what's happening at the gut mucosa, but it's medically not possible at this time to do that. But as Gordon, as you state, in mice, you do see regional differences in the fungal communities.
Okay, so we've got a couple more questions before I hand you back to Oliver, which is again related. It's about the PDC part of the story. So yeah. It's rare. So Peter Cook has mentioned they're a rare population. And have you looked at the locational aspects of these interactions? And the other question again, which is related, do they migrate to the lymph nodes with large chunks of aspergillus hyphae, or is it all done in the local environment? So in other words, the location of these cells, is that important?
Well, I hope I get Peter Cook as a reviewer for my NIH grant, because I just submitted it 10 days ago, my 5-year renewal, and this is sort of one aim of our renewal.
He's a very tough reviewer, Tobias.
Yeah. So that is something we're actively looking at right now. The challenge has been really to define good staining protocols to identify PDCs in the lung. And we don't have a good mouse model in which, with a PDC-CRE, for example, where we could drive a fluorescent protein that would unambiguously identify PDCs. But I think we'll get there. I think Peter's question is highly relevant because PDCs are known to form synapses with virally infected cells via integrins and ICAMs. And so, the question of whether you need cell-to-cell contact for activation versus whether this is entirely cytokine driven, I think these are models that need to be distinguished rigorously. Based on the number of PDCs we see in the lung, I favor the latter model, that it's a cytokine driven process, simply because we have 100 neutrophils for every PDC in the lung. And it would be hard to imagine that a PDC could come and just, interact very, very rapidly with all these neutrophils. And we don't know yet if they carry antigen to the lung draining lymph node.
Well, it could be in localized clusters, I guess. But...
Yeah, no, absolutely. And we need to do the experiments.
Okay, thank you. So for the last couple of questions, I notice we're running out of time. I'll hand you back to Oliver for a last couple of questions.
Right. So we have a few remaining questions, and one is whether in the epimutants that you analyzed, Joe, you saw any differences with regard to metabolic features?
Good question. I don't know that we've explored that in sufficient detail. We did test the epimutants in FKBP12 that formed hyphae to at least test whether or not they were locked in the hyphal phase, and they can still respond to hypoxia and revert to being a yeast. But I think there's a lot more that could potentially be done with metabolomics. But with respect to FKBP12, there's not a lot of work implicating it in metabolism of the cell. There is a really beautiful paper published in PLOS Genetics recently showing that it's involved in regulating transcription factors that regulate ribosomal protein expression. And that may explain why there's a growth defect in cells lacking FKBP12. And that might have a lot of metabolic consequences from reduced translation capacity.
Okay, if you analyze your RNAi deficient strains, did you observe any additional mutations or effects accumulating in the genomes of these strains? For example, suggested here transposon insertions.
That's an excellent question, because a prime role for the RNAi pathway, one is to control double-strand RNA mycoviruses. The other is to constrain and control transposable elements. And in our previous work in Cryptococcus, if we cross RNAi mutants, you find all sorts of transposition occurring in the progeny and a very large mutational burden. We haven't published it yet, but we've also been looking at transposition in Mucor and found a high rate of transposon insertion into the FKBP12 locus. In one case, that was as a consequence of infection. In the other cases, it's in the RNAi mutant backgrounds. So a very astute question, and I think there'll be more to come on that theme and topic.
Okay, so I've been threatened in my private chat by another person here in the audience to stop asking questions. So I'll hand back to that person and have him give the closing remarks. Gordon.
Thank you, Oliver. Yes, I can't rugby tackle you off the stage, but I can certainly do something to make my feelings known. Thank you. Well, I want to just close by thanking Joe and Tobias. Thank you so much for such wonderful talks. It was really great. It's really nice to hear all this exciting research, and to thank the audience, the attendees, for coming to this session and for all your wonderful questions. And I wish you all health and happiness, and we will see you in 1 month's time. Goodbye.
Thank you.